Hello everyone, welcome back. We are starting into chapter 3 here, which will be discussing cell biology concepts. Since we are cellular organisms, everything we will understand really in physiology, less so in anatomy, will be centered around cells and how they function and how they work. And so that's why we have a whole chapter dedicated to that. So let's get started. So question here, what are the main parts of the cell? Try to think of them. There's a number of them, and some of you may have had this in high school, but the main ones to note that we'll be focusing on is the nucleus, right? So the nucleus contains all of the DNA, genomic information, regulates gene expression, right? So it's an important organelle here. We have mitochondria, right? The classic statement of mitochondria is the powerhouse of the cell. It's where most of the ATP is formed, right, for energy production in the cell. So mitochondria, also organelle bound. We have the Golgi apparatus, right, that packages and transports proteins and other biomolecules throughout the cell, right, delivering them where they need to go. We have the endoplasmic reticulum and the rough endoplasmic reticulum. We use the acronym ER here, so rough ER and smooth ER, right? And this is where proteins are synthesized as well as some other biomolecules, right? So this is kind of like the factory of the cell. We have the plasma membrane, which is an important part of the cell, right? It keeps everything out, and everything in. So it's like the wall around a city. And we have the cytoplasm. This is kind of the liquid suspension fluid that suspends all of the organelles and other molecules and components and particles in the cell. And then we have ribosomes, which are enzymes, right? And the ribosomes actually make the protein. So those kind of most important parts of the cell that we'll reference here. So we kind of have, here's an overview of what we'll go through, the picture of the cell. And this is in your textbook again. I like to use figures from the textbook. Right, so we have our nucleus, which is the big purple blob looking thing. We have our mitochondria. We have our Golgi apparatus. We have vesicles and transport bubbles, and they kind of look like here. We have our cytoplasm, which is the fluid in between. We have our ER and rough ER and smooth ER. We have our plasma membrane keeping everything in and everything else out, right? So this is just a nice overview of physically representing what the cell is. So we'll kind of start from the outside in here. So we'll actually start with the plasma membrane. So the main functions of the plasma membrane, really the main thing is it's a boundary, right? Separating the cytoplasm, so inside the cell, or intracellular, intra means inside, and cell, right? Keeps things that's supposed to be inside the cell, keeps them inside, and everything that's supposed to be outside. It supports and encloses the cell content, so it provides kind of a physical structure for the cell. One thing it does is it helps the cell attach to its external environment, because there's proteins that protrude out of the cell membrane or plasma membrane that we'll kind of highlight a little bit, not too much, but it helps the cell connect to its environment. It helps communicate and recognize. Again, there's another class of proteins that stick out of the cell membrane that are receptors, right, as well as communicators to other cells surrounding it. And then one big thing, too, that the plasma membrane does is it is semi-permeable or selectively permeable in the sense that it can regulate what goes in and what goes out of the cell, right? And that's a big function that the cell does, because you don't want unwanted material coming in, and you don't want your precious material going out. So the plasma membrane, though it's seemingly quite simple, it's pretty important. It does have a unique composition here. So it is a phospholipid bilayer. Remember from chapter 2 we talked about lipids. Phospholipids was one of them, right? So they are amphilipids, having a polar head and a nonpolar tail, right? And this two of those phospholipids, stick together where the nonpolar tails touch and then the polar heads stick out towards the water, right? You may think, well, why is the head pointing out? That makes sense because there's water outside the cell. But why is the polar head pointing in? Well, that's because the inside of the cell has lots of water in it. So that's how we create our phospholipid bilayer, where, the inside is nonpolar, liking more oil-like substances, and then you have your polar head that is hydrophilic, right? It loves water, right? There's other molecules and proteins inside this, creating what's called the fluid mosaic model. These proteins and molecules can just float through the membrane like a raft. It's more organized and structured than that, but that's the model that's been held for a long time. One example here that the textbook provides is the glycocalyx. This is a combination of carbohydrates and lipids, so glycolipids and proteins, glycoproteins. These molecules kind of hybridize together, as you can see here, right? And this should be like a glycoprotein here. It helps communicate with cells surrounding this cell, or it helps, it bind and identify other molecules outside the cell. So like I said, there's a phospholipid bilayer for polar heads facing outside and inside where the water is located. Then the nonpolar tails are facing on the inside. There is also cholesterol, right? And this is interspersed. So remember, this is also a lipid, but its chemical structure is more ring-based than linear. It helps determine how fluid the membrane is, right? So versus stiff, basically you can view it stiff versus soft. More cholesterol, the softer it is. So cholesterol is an important part of the cell. And then, of course, I mentioned the fluid mosaic model, right? Distribution of all these molecules in the membrane move back and forth, kind of randomly. At least we think randomly, probably not. At least that's what the science is starting to point out. Nothing is ever random in biology, as much as they tried to convince us as such, and then they're surprised when it's not random. So take that with a grain of salt when that comes up in other textbooks. There are the membrane proteins, right? So like I said, there are molecules and proteins embedded in this plasma membrane or cell membrane. They can be integral membrane proteins, or they extend deeply into the plasma membrane. Often this is connecting like the outside surface to the inside surface. So it's bridging between both phospholipid layers. This allows us to form one example of channels through the membrane. So some molecules are too big to diffuse or move freely through the plasma membrane. So you need something or some way to get them through. One of them is a channel, which is vitally important for muscle cells and neurons, right? So these channels we're actually going to talk a lot about in this chapter, as well as when we talk about them in the muscle skeletal chapters, which I think is 9 and 10, and then our neuron cells, which will be in the spring semester. There is also peripheral membrane proteins. These stick to kind of like the inner and outer surfaces of the lipid layer, and these are more related to signaling purposes. So they don't stick all the way through the plasma membrane. That's what this is peripheral membranes trying to show, because they have a different function, which is more of like an antenna or receptor of receiving or transmitting a chemical signal to communicate either outside the cell or inside the cell to tell the cell what to do or other cells what to do, right? Which is important. Cells need other cells to survive. That's a very important concept in cellular biology. And the only way to really sense if you're alone or you have neighbors is through these like peripheral membrane proteins, right? So again, like all proteins here, the 3D shape determines the function of the proteins. So it also determines some chemical characteristics, which we're not going to be too concerned about here. But the shape of the protein determines the function, and that actually helps scientists understand what a function is. If it's a new protein, we can just start looking and investigating its shape and be like, oh, well, this kind of protein looks like a channel protein, or it looks like an enzyme, right? So there's a lot of different shapes that are common throughout biology for proteins, and this functionality of their shape helps us identify them. So one important thing that, again, we need to really highlight here are transport proteins. So transport proteins are proteins that are located in the plasma membrane, the phospholipid bilayer, right? They are, so we go back one slide here, they are integral membrane proteins, so they span both, as we can see here, right? Span both the inner and the outer, depending on your viewpoint. But they allow certain molecules and elements to move through the membrane freely or regulated, which is probably more important. But there's kind of three main classes that we'll look at. Channel proteins kind of form a channel, which just kind of like, it's like the Panama Canal, really. Things can move through, typically in one direction, but there's not really much regulation going on at this level. There are carrier proteins, which actually have kind of like an enzyme, a lock and key model or induced fit model, but they're specifically designed to only transport one molecule or protein or anything, one of those things, with a very specific shape through the plasma membrane. Again, typically in one direction. Sometimes they can go forwards and backwards like a reversible pump, but we're not going to worry about that too much right now. But there are carrier proteins. Then there are ATP-powered pumps. Sometimes you need a little extra boost to move molecules through from one side to the next. And there are protein pumps that use ATP. Remember, ATP is the power, the battery pack for the cell. It's the energy source. It can use ATP, breaking it into ADP to move molecules across the membrane. This is typically used when you're moving a molecule to a place where it doesn't want to go, i.e. against a concentration gradient. That's a big word, and I'm introducing that here and now because it's going to come up here in this chapter. So those are the three types of transport molecules there. So transport proteins, channel proteins, there's a whole bunch of different kinds of them, and really we kind of name them based upon what they are channeling, right? So you can have channel proteins, they're integral membrane proteins. They are formed little itty-bitty channels from the membrane. And the size of the channel determines what moves through, right? And I also mentioned when we talked about chemistry in chapter 2, which is the last chapter, right? These ion charges, the positive or the negative charges, is important to note. And this is where it's going to come up, is because these charges is one thing that these channels are also watching for, right? So the shape and the size and the charge of what molecules are trying to move through determine what channel it's going to move through. So like we have a potassium leak channel, right? So this one's always open, right? So it's leaking potassium. You can have a ligand-gated sodium channel, which is closed at the moment. But then when a ligand or a very specific signaling molecule, that's what this fancy ligand word means, it's just a signal molecule, touches the channel, it opens up and sodium now can move through. So these proteins have different parts to them. I'm not going to get too lost in the weeds with the hydrophobic and hydrophilic regions, but if you think about it, there are hydrophobic regions that are inside, right, in the middle of this plasma membrane, and then there's hydrophilic, which love, the water, that are outside, right, and then line the channel there, or the tunnel. And then the charge of the proteins, or basically the charge of the tunnel, helps identify what ions can move through and what can't move through. So that's the channel proteins here. The ion-gated leak ion channels here, right? This is largely responsible for a lot of the permeability of the plasma membrane. When it comes to like muscle movement, this really comes into effect. We'll really talk about this, but this is constantly open, and it's leaking like a water faucet on a slow drip, right? It's always open, always moving through, letting potassium move through. Then there's the ligand-gated, which responds to some sort of stimulus, typically a signal, and opens up. And then there's also this voltage gauge ion channel, right? So this is important in muscle cells and in neuron cells, right? So a voltage or a charge can open and close channels. Moving through, right, so we can have these transport proteins or carrier proteins. They're transporters because what they're doing is they're physically moving larger molecules, excluding like small ions and stuff. It's really more involved in moving large molecules, right? So they can move only specific molecules, right? Which then they enter the carrier, step one. Then they bind to some site, right? And then the molecule releases it into the other side, right? So there's different types of carrier proteins. There's uniporters, so they only move one molecule, right? So only identifies one and only moves one, right? There's symporters that can move two molecules in the same direction. And then there's antiporters. So one molecule moves down, the other molecule moves up, so it kind of like transfers molecules. One comes in, one goes out, kind of a sort of thing. And all of these transporter proteins are important in cell biology as well. So then we have our ATP-powered pumps, right? So they require ATP to move whatever they're trying to move, right? So it breaks it apart. So remember ATP, you break it into ADP, it releases energy. That's what's going on here via a reaction, just called a hydrolysis reaction. Don't worry about the title of it, but under a reaction, right? And it moves this example here is like I would think it's an ion, but it looks more like a sodium one for me. It moves it across the membrane. And typically, like I said, this is used to move molecules against the concentration gradient. So typically, we'll talk about this probably in part 2 here, but molecules love to go from high concentration areas to low concentration areas. But what if the cell needs it to go the opposite direction, from low to high, right? So it's like people packed in there's 2 rooms. When you live in we'll use a dorm example. Your roommate or your neighbors in the dorm room next to you, they're having a party, right? It's jam-packed with 30 people. It's hopping, right? Well, some people are going to think, oh, that room's too clustered, too many people. They're going to move to yours, right? Because you just have yourself and your roommate, right? So then people start moving to your room because, it's easier to get into, right? But what if you don't want them in your room? You push them back into your neighbor's room, go back and have a party over there, right? It takes a lot of energy to move them back into their party room, right? That's kind of what these ATP power pumps are doing. They're moving something from a low to a high concentration. Hopefully that metaphor makes sense. When you're seniors at ORU, you'll probably experience something like that. I did when I was at ORU, so parties are fun unless you're trying to go to sleep. There are other types of membrane proteins here. They are receptor proteins. They basically receive a signal, which is called a ligand, right? Just signal, ligand, you can think that the same, right? These ligands bind to very specific proteins and molecules in the plasma membrane, right? Lock and key model, that's a hard, fast set rule. There's no changing that rule, right? They bind to a receptor site that sticks outside the cell, right? Because that signal from outside the cell binds to these receptors, it's like an antennae, and then it changes a receptor protein in the plasma membrane to tell the cell, go do something, right? Example of this kind of receptor protein is a G protein coupled receptors. GPCRs is the acronym for them. I don't think we'll worry about them too much, but I'm just giving you an example of what that can be. They can also be bound to transport proteins, right? So it's kind of an interesting situation. Don't really want to get lost too much here. We'll hit a number of these concepts again in other chapters with better specific examples, but I just want to introduce it here. There are also enzyme proteins bound in the membrane, right? And so these are going to be catalyzing very specific reactions An example of this is in our digestive system, right? There's going to be enzymes bound on the epithelial cells lining your small intestine that will digest peptide bonds, right? Breaking down the proteins that you're eating into the basic amino acids, right? Oh, wow. That's why we learn about amino acids and peptides, right? So that's an example. Like, there's a lot of different proteins in your plasma membrane, and there's those three main classes: transports, right? Receptors, and then enzyme proteins. So we'll pause this video here and we'll move on to the next section.